Solvent. Although many weak bases, 2.2 Ionization of water, weak acids, and weak bases. Although many of the solvent properties of water can be explained in terms of the uncharged H2O molecule, the small degree of ionization of water to hydrogen ions, H+, and hydroxide ions, OH, must also be taken into account. Like all reversible reactions, the ionization of water can be described by an equilibrium constant. When weak acids are dissolved in water, they contribute H+ by ionizing. Weak bases consume H+ by becoming protonated. These processes are also governed by equilibrium constants. The total hydrogen ion concentration from all sources is experimentally measurable and is expressed as the pH of the solution. To predict the state of ionization of solutes in water, we must take into account the relevant equilibrium constants for each ionization reaction. We therefore turn now to a brief discussion of the ionization of water and of weak acids and bases dissolved in water. Pure water is slightly ionized. Water molecules have a slight tendency to undergo reversible ionization to yield a hydrogen ion, a proton, and a hydroxide ion, giving the equilibrium. Although we commonly show the dissociation product of water as H+, free protons do not exist in solution. Hydrogen ions formed in water are immediately hydrated to form hydronium ions, H3O+. Hydrogen bonding between water molecules makes the hydration of dissociating protons virtually instantaneous. The ionization of water can be measured by its electrical conductivity. Pure water carries electrical current as H3O+ migrates toward the cathode and OH toward the anode. The movement of hydronium and hydroxide ions in the electric field is extremely fast compared with that of other ions such as Na+, K+, and Cl. This high ionic mobility results from the kind of proton hopping shown in Figure 2-14. No individual proton moves very far through the bulk solution, but a series of proton hops between hydrogen-bonded water molecules causes the net movement of a proton over a long distance in a remarkably short time. OH also moves rapidly by proton hopping, but in the opposite direction. As a result of the high ionic mobility of H+, acid-base reactions in aqueous solutions are exceptionally fast. As noted above, proton hopping very likely also plays a role in biological proton transfer reactions, Fig. 2-10, see also Fig. 20-29b. Because reversible ionization is crucial to the role of water in cellular function, we must have a means of expressing the extent of ionization of water in quantitative terms. A brief review of some properties of reversible chemical reactions shows how this can be done. The position of equilibrium of any chemical reaction is given by its equilibrium constant, Keq, sometimes expressed simply as K. For the generalized reaction, the equilibrium constant Keq can be defined in terms of the concentrations of reactants, A and B, and products, C and D, at equilibrium. Figure 2-14 Proton hopping. Short hops of protons between a series of hydrogen-bonded water molecules result in an extremely rapid net movement of a proton over a long distance. As a hydronium ion, upper left, gives up a proton, a water molecule some distance away, bottom, acquires one, becoming a hydronium ion. Proton hopping is much faster than true diffusion and explains the remarkably high ionic mobility of H+ ions compared with other monovalent cations such as Na+ and K+. Strictly speaking, the concentration terms should be the activities, or effective concentrations in non-ideal solutions, of each species. Except in very accurate work, however, the equilibrium constant may be approximated by measuring the concentrations at equilibrium. For reasons beyond the scope of this discussion, equilibrium constants are dimensionless. Nonetheless, we have generally retained the concentration units m in the equilibrium expressions used in this book to remind you that molarity is the unit of concentration used in calculating Keq. The equilibrium constant is fixed and characteristic for any given chemical reaction at a specified temperature. It defines the composition of the final equilibrium mixture, regardless of the starting amounts of reactants and products. Conversely, we can calculate the equilibrium constant for a given reaction at a given temperature if the equilibrium concentrations of all its reactants and products are known. As we showed in Chapter 1, the standard free energy change, delta G degree, is directly related to ln Keck. The ionization of water is expressed by an equilibrium constant. The degree of ionization of water at equilibrium, EC 2 to 1, is small. At 25 degrees Celsius only about 2 of every 10 9 molecules in pure water are ionized at any instant. The equilibrium constant for the reversible ionization of water is: in pure water at 25 degrees Celsius, the concentration of water is 55.5 m grams of H2O and 1 L divided by its gram molecular weight, 1000 G/L/18.015 grams per mole, and is essentially constant in relation to the very low concentrations of H+ and OH, namely 1 × 10⁷ m. Accordingly, we can substitute 55.5 m in the equilibrium constant expression, EC 2 to 3, to yield. On rearranging, this becomes: where Kw designates the product, 55.5 m Keck, the ion product of water at 25 degrees Celsius. The value for Keck, determined by electrical conductivity measurements of pure water, is 1.8 × 1016 m at 25 degrees Celsius. Substituting this value for Keck in equation 2 to 4 gives the value of the ion product of water. Thus the product, H+, OH, in aqueous solutions at 25 degrees Celsius always equals 1 × 1014 m2. When there are exactly equal concentrations of H+ and OH, as in pure water, the solution is said to be at neutral pH. At this pH, the concentrations of H+ and OH can be calculated from the ion product of water as follows. Solving for H+ gives: as the ion product of water is constant, whenever H+ is greater than 1 × 10⁷ m, OH must be less than 1 × 10⁷ m, and vice versa. When H+ is very high, as in a solution of hydrochloric acid, OH must be very low. From the ion product of water we can calculate H+, if we know OH, and vice versa. Worked example 2 to 3 calculation of H+. What is the concentration of H+ in a solution of 0.1 m Na OH? Solution: we begin with the equation for the ion product of water. With OH equals 0.1 m, solving for H+ gives. Worked example 2 to 4 calculation of OH. What is the concentration of OH in a solution with an H+ concentration of 1.3 × 10⁴ m? Solution: we begin with the equation for the ion product of water. With H+ equals 1.3 × 10⁴ m, solving for OH gives. In all calculations be sure to round your answer to the correct number of significant figures, as here. The pH scale designates the H+ and OH concentrations. The ion product of water, Kw, is the basis for the pH scale, table 2 to 6. It is a convenient means of designating the concentration of H+, and thus of OH, in any aqueous solution in the range between 1.0 m H+ and 1.0 m OH. The term pH is defined by the expression table 2 to 6. The pH scale H+, the symbol P denotes negative logarithm of. For a precisely neutral solution at 25 degrees Celsius, in which the concentration of hydrogen ions is 1.0 × 10⁷ m, the pH can be calculated as follows. Note that the concentration of H+ must be expressed in molar, m terms. The value of 7 for the pH of a precisely neutral solution is not an arbitrarily chosen figure, it is derived from the absolute value of the ion product of water at 25 degrees Celsius, which by convenient coincidence is a round number. Solutions having a pH greater than 7 are alkaline or basic. The concentration of OH is greater than that of H+. Conversely, solutions having a pH less than 7 are acidic. Keep in mind that the pH scale is logarithmic, not arithmetic. To say that two solutions differ in pH by 1 pH unit means that one solution has 10 times the H+ concentration of the other, but it does not tell us the absolute magnitude of the difference. Figure 2-15 gives the pH values of some common aqueous fluids. A cola drink, pH 3.0, or red wine, pH 3.7, has an H+ concentration approximately 10,000 times that of blood, pH 7.4. The pH of an aqueous solution can be approximately measured with various indicator dyes, including litmus, phenolphthalein, and phenol red. These dyes undergo color changes as a proton dissociates from the dye molecule. Accurate determinations of pH in the chemical or clinical laboratory are made with a glass electrode that is selectively sensitive to H+. Concentration but insensitive to Na+, K+, and other cations. In a pH meter, the signal from the glass electrode placed in a test solution is amplified and compared with the signal generated by a solution of accurately known pH. Figure 2-15 The pH of some aqueous fluids. Measurement of pH is one of the most important and frequently used procedures in biochemistry. The pH affects the structure and activity of biological macromolecules. For example, the catalytic activity of enzymes is strongly dependent on pH. See Fig. 2-22. Measurements of the pH of blood and urine are commonly used in medical diagnoses. The pH of the blood plasma of people with severe, uncontrolled diabetes, for example, is often below the normal value of 7.4. This condition is called acidosis, described in more detail below. In certain other diseases, the pH of the blood is higher than normal, a condition known as alkalosis. Extreme acidosis or alkalosis can be life-threatening. Weak acids and bases have characteristic acid dissociation constants. Hydrochloric, sulfuric, and nitric acids, commonly called strong acids, are completely ionized in dilute aqueous solutions. The strong bases NaOH and KOH are also completely ionized. Of more interest to biochemists is the behavior of weak acids and bases, those not completely ionized when dissolved in water. These are ubiquitous in biological systems and play important roles in metabolism and its regulation. The behavior of aqueous solutions of weak acids and bases is best understood if we first define some terms. Acids may be defined as proton donors and bases as proton acceptors. When a proton donor such as acetic acid, CH3COOH, loses a proton, it becomes the corresponding proton acceptor, in this case the acetate anion, CH3COO. A proton donor and its corresponding proton acceptor make up a conjugate acid-base pair, Fig. 2-16, related by the reversible reaction. Figure 2-16 conjugate acid-base pairs consist of a proton donor and a proton acceptor. Some compounds, such as acetic acid and ammonium ion, are monoprotic, they can give up only one proton. Others are diprotic, carbonic acid and glycine, or triprotic, phosphoric acid. The dissociation reactions for each pair are shown where they occur along a pH gradient. The equilibrium or dissociation constant Ka, and its negative logarithm, the pKa, are shown for each reaction. Asterisk for an explanation of apparent discrepancies in pKa values for carbonic acid, H2CO3, Cp. 67. Each acid has a characteristic tendency to lose its proton in an aqueous solution. The stronger the acid, the greater its tendency to lose its proton. The tendency of any acid, HA, to lose a proton and form its conjugate base, is defined by the equilibrium constant, Keq, for the reversible reaction. For which equilibrium constants for ionization reactions are usually called ionization constants or acid dissociation constants, often designated Ka. The dissociation constants of some acids are given in Figure 2-16. Stronger acids, such as phosphoric and carbonic acids, have larger ionization constants. Weaker acids, such as monohydrogen phosphate, have smaller ionization constants. Also included in Figure 2-16 are values of pKa, which is analogous to pH, and is defined by the equation. The stronger the tendency to dissociate a proton, the stronger is the acid, and the lower its pKa. As we shall now see, the pKa of any weak acid can be determined quite easily. Titration curves reveal the pKa of weak acids. Titration is used to determine the amount of an acid in a given solution. A measured volume of the acid is titrated with a solution of a strong base, usually sodium hydroxide, NaOH, of known concentration. The NaOH is added in small increments until the acid is consumed, neutralized, as determined with an indicator dye or a pH meter. The concentration of the acid in the original solution can be calculated from the volume and concentration of NaOH added. The amounts of acid and base in titrations are often expressed in terms of equivalence, where one equivalent is the amount of a substance that will react with, or supply, 1 mole of hydrogen ions in an acid-base reaction. A plot of pH against the amount of NaOH added, a titration curve, reveals the pKa of the weak acid. Consider the titration of a 0.1 m solution of acetic acid with 0.1 m NaOH at 25 degrees Celsius, Fig. 2-17. Two reversible equilibria are involved in the process. Here, for simplicity, acetic acid is denoted HAC. The equilibria must simultaneously conform to their characteristic equilibrium constants, which are, respectively. At the beginning of the titration, before any NaOH is added, the acetic acid is already slightly ionized, to an extent that can be calculated from its ionization constant, Ec 2-8. Figure 2-7. As NaOH is gradually introduced, the added OH combines with the free H+ in the solution to form H2O, to an extent that satisfies the equilibrium relationship in equation 2-7. As free H+ is removed, HAC dissociates further to satisfy its own equilibrium constant, Ec 2-8. The net result as the titration proceeds is that more and more HAC ionizes, forming AC, as the NaOH is added. At the midpoint of the titration, at which exactly 0.5 equivalent of NaOH has been added per equivalent of the acid, 1/2 of the original acetic acid has undergone dissociation, so that the concentration of the proton donor, HAC, now equals that of the proton acceptor, AC. At this midpoint a very important relationship holds: the pH of the equimolar solution of acetic acid and acetate is exactly equal to the pKa of acetic acid, pKa equals 4.76, Figs 2-16, 2-17. The basis for this relationship, which holds for all weak acids, will soon become clear. As the titration is continued by adding further increments of NaOH, the remaining non-dissociated acetic acid is gradually converted into acetate. The end point of the titration occurs at about pH 7.0. All the acetic acid has lost its protons to OH, to form H2O and acetate. Throughout the titration the two equilibria, Equans 2-5, 2-6, coexist, each always conforming to its equilibrium constant. Figure 2-8. The regions of buffering capacity. Figure 2-18 compares the titration curves of three weak acids with very different ionization constants. Acetic acid, pKa equals 4.76. Dihydrogen phosphate, pKa equals 6.86, and ammonium ion, pKa equals 9.25. Although the titration curves of these acids have the same shape, they are displaced along the pH axis because the three acids have different strengths. Acetic acid, with the highest Ka, lowest pKa, of the three, is the strongest of the three weak acids, loses its proton most readily, it is already half dissociated at pH 4.76. Dihydrogen phosphate loses a proton less readily, being half dissociated at pH 6.86. Ammonium ion is the weakest acid of the three, and does not become half dissociated until pH 9.25. The titration curve of a weak acid shows graphically that a weak acid and its anion a conjugate acid-base pair can act as a buffer, as we describe in the next section. Summary 2.2 Ionization of water, weak acids and weak bases. Pure water ionizes slightly, forming equal numbers of hydrogen ions, hydronium ions, H3O+, and hydroxide ions. The extent of ionization is described by an equilibrium constant from which the ion product of water, Kw, is derived. At 25 degrees Celsius, Kw equals H plus, OH equals 55.5 m, Kek equals 1014 m2. The pH of an aqueous solution reflects, on a logarithmic scale, the concentration of hydrogen ions. The greater the acidity of a solution, the lower its pH. Weak acids partially ionize to release a hydrogen ion, thus lowering the pH of the aqueous solution. Weak bases accept a hydrogen ion, increasing the pH. The extent of these processes is characteristic of each particular weak acid or base and is expressed as an acid dissociation constant. The pKa expresses, on a logarithmic scale, the relative strength of a weak acid or base. The stronger the acid, the smaller its pKa, the stronger the base, the larger its pKa. The pKa can be determined experimentally. It is the pH at the midpoint of the titration curve for the acid or base. 2.3 Buffering against pH changes in biological systems. Almost every biological process is pH dependent. A small change in pH produces a large change in the rate of the process. This is true not only for the many reactions in which the H+ ion is a direct participant, but also for those reactions in which there is no apparent role for H+ ions. The enzymes that catalyze cellular reactions, and many of the molecules on which they act, contain ionizable groups with characteristic pKa values. The protonated amino and carboxyl groups of amino acids, and the phosphate groups of nucleotides, for example, function as weak acids. Their ionic state is determined by the pH of the surrounding medium. When an ionizable group is sequestered in the middle of a protein, away from the aqueous solvent, its pKa, or apparent pKa, can be significantly different from its pKa in water. As we noted above, ionic interactions are among the forces that stabilize a protein molecule and allow an enzyme to recognize and bind its substrate. Cells and organisms maintain a specific and constant cytosolic pH, usually near pH 7, keeping biomolecules in their optimal ionic state. In multicellular organisms, the pH of extracellular fluids is also tightly regulated. Constancy of pH is achieved primarily by biological buffers, mixtures of weak acids, and their conjugate bases. Buffers are mixtures of weak acids and their conjugate bases. Buffers are aqueous systems that tend to resist changes in pH when small amounts of acid, H+, or base, OH, are added. A buffer system consists of a weak acid, the proton donor, and its conjugate base, the proton acceptor. As an example, a mixture of equal concentrations of acetic acid and acetate ion, found at the midpoint of the titration curve in Figure 2-17, is a buffer system. Notice that the titration curve of acetic acid has a relatively flat zone extending about 1 pH unit on either side of its midpoint pH of 4.76. In this zone, a given amount of H+ or OH added to the system has much less effect on pH than the same amount added outside the zone. This relatively flat zone is the buffering region of the acetic acid-acetate buffer pair. At the midpoint of the buffering region, where the concentration of the proton donor, acetic acid, exactly equals that of the proton acceptor, acetate, the buffering power of the system is maximal, that is, its pH changes least on addition of H+ or OH. The pH at this point in the titration curve of acetic acid is equal to its pKa. The pH of the acetate buffer system does change slightly when a small amount of H+ or OH is added, but this change is very small compared with the pH change that would result if the same amount of H+ or OH were added to pure water or to a solution of the salt of a strong acid and strong base, such as NaCl, which has no buffering power. Buffering results from two reversible reaction equilibria occurring in a solution of nearly equal concentrations of a proton donor and its conjugate proton acceptor. Figure 2 to 19 explains how a buffer system works. Whenever H plus or OH is added to a buffer, the result is a small change in the ratio of the relative concentrations of the weak acid and its anion, and thus a small change in pH. The decrease in concentration of one component of the system is balanced exactly by an increase in the other. The sum of the buffer components does not change, only their ratio changes. Each conjugate acid-base pair has a characteristic pH zone in which it is an effective buffer, Fig. 2 to 18. The pair has a pKa of 6.86 and thus can serve as an effective buffer system between approximately pH 5.9 and pH 7.9. The pair, with a pKa of 9.25, can act as a buffer between approximately pH 8.3 and pH 10.3. The Henderson-Hasselbalch equation relates pH, pKa, and buffer concentration. The titration curves of acetic acid and Fig. 2 to 18 have nearly identical shapes, suggesting that these curves reflect a fundamental law or relationship. This is indeed the case. The shape of the titration curve of any weak acid is described by the Henderson-Hasselbalch equation, which is important for understanding buffer action and acid-base balance in the blood and tissues of vertebrates. This equation is simply a useful way of restating the expression for the ionization constant of an acid. For the ionization of a weak acid HA, the Henderson-Hasselbalch equation can be derived as follows. Figure 2 to 19. The acetic acid acetate pair as a buffer system. The transiently restore the product. First solve for H+. Then take the negative logarithm of both sides. Substitute pH for log H+ and pKa for log Ka. Figure 2 to 20. Ionization of histidine. The amino acid histidine. Now invert log HA / which involves changing its sign, to obtain the Henderson-Hasselbalch equation. This equation fits the titration curve of all weak acids, and enables us to deduce some important quantitative relationships. For example, it shows why the pKa of a weak acid is equal to the pH of the solution at the midpoint of its titration. At that point, HA equals A and pH equals pKa plus log 1 equals pKa plus 0 equals pKa. The Henderson-Hasselbalch equation also allows us to 1. Calculate pKa, given pH, and the molar ratio of proton donor and acceptor. 2. Calculate pH, given pKa and the molar ratio of proton donor and acceptor, and 3. Calculate the molar ratio of proton donor and acceptor, given pH and pKa. Weak acids or bases buffer cells and tissues against pH changes. The intracellular and extracellular fluids of multicellular organisms have a characteristic and nearly constant pH. The organism's first line of defense against changes in internal pH is provided by buffer systems. The cytoplasm of most cells contains high concentrations of proteins, and these proteins contain many amino acids with functional groups that are weak acids or weak bases. For example, the side chain of histidine, Fig. 2 to 20, has a pKa of 6.0 and thus can exist in either the protonated or unprotonated form near neutral pH. Proteins containing histidine residues therefore buffer effectively near neutral pH. Worked example. Calculate the substituting pK2 equal. This gives us nucleotides such as ATP, as well as many metabolites of low molecular weight, contain ionizable groups that can contribute buffering power to the cytoplasm. Some highly specialized organelles and extracellular compartments have high concentrations of compounds that contribute buffering capacity. Organic acids buffer the vacuoles of plant cells, ammonia buffers urine. Two especially important biological buffers are the phosphate and bicarbonate systems. The phosphate buffer system, which acts in the cytoplasm of all cells, consists of as proton donor and as proton acceptor. The phosphate buffer system is maximally effective at a pH close to its pKa of 6.86, Figs 2 to 16, 2 to 18, and thus tends to resist pH changes in the range between about 5.9 and 7.9. It is therefore an effective buffer in biological fluids. In mammals, for example, extracellular fluids and most cytoplasmic compartments have a pH in the range of 6.9 to 7.4. Worked A. Solution. In this case, we can roughly check this. B. If 1. Solution. C. If solution. So Po equals. Blood plasma is buffered in part by the bicarbonate system, consisting of carbonic acid as proton donor and bicarbonate, HCO, as proton acceptor. K1 is the first of several equilibrium constants in the bicarbonate buffering system. This buffer system is more complex than other conjugate acid-base pairs, because one of its components, carbonic acid, H2CO3, is formed from dissolved carbon dioxide and water in a reversible reaction. Carbon dioxide is a gas under normal conditions, and CO2 dissolved in an aqueous solution is in equilibrium with CO2 in the gas G phase. The pH of a bicarbonate buffer system depends on the concentration of H2CO3 and the proton donor and acceptor components. The concentration of H2CO3 in turn depends on the concentration of dissolved CO2, which in turn depends on the concentration of CO2 in the gas phase, or the partial pressure of CO2, denoted PCO2. Thus the pH of a bicarbonate buffer exposed to a gas phase is ultimately determined by the concentration of in the aqueous phase and by PCO2 in the gas phase. The bicarbonate buffer system is an effective physiological buffer near pH 7.4, because the H2CO3 of blood plasma is in equilibrium with a large reserve capacity of CO2 G in the air space of the lungs. As noted above, this buffer system involves three reversible equilibria, in this case between gaseous CO2 in the lungs and bicarbonate in the blood plasma, Fig. 2-21. Blood can pick up H+, such as from the lactic acid produced in muscle tissue during vigorous exercise. Alternatively, it can lose H+, such as by protonation of the NH3 produced during protein catabolism. When H+ is added to blood as it passes through the tissues, reaction 1 in Figure 2-21 proceeds toward a new equilibrium in which H2CO3 is increased. This in turn increases CO2 in the blood, reaction 2, and thus increases the partial pressure of CO2 G in the airspace of the lungs, reaction 3. The extra CO2 is exhaled. Conversely, when H+ is lost from the blood, the opposite events occur. More H2CO3 dissociates into H+, and thus more CO2 G from the lungs dissolves in blood plasma. The rate of respiration, that is, the rate of inhaling and exhaling, can quickly adjust these equilibria to keep the blood pH nearly constant. The rate of respiration is controlled by the brain stem, where detection of an increased blood PCO2 or decreased blood pH triggers deeper and more frequent breathing. Figure 2-21. Hyperventilation, the rapid breathing sometimes elicited by stress or anxiety, tips the normal balance of O2 breathed in and CO2 breathed out in favor of too much CO2 breathed out, raising the blood pH to 7.45 or higher. This alkalosis can lead to dizziness, headache, weakness, and fainting. One home remedy for mild alkalosis is to breathe briefly into a paper bag. The air in the bag becomes enriched in CO2, and inhaling this air increases the CO2 concentration in the body and blood and decreases blood pH. At the normal pH of blood plasma, 7.4, very little H2CO3 is present. Relative to, and the addition of just a small amount of base, NH3 or OH, would titrate this H2CO3, exhausting the buffering capacity. The important role of H2CO3, pKa equals 3.57 at 37 degrees Celsius, in buffering blood plasma pH. 7.4 seems inconsistent with our earlier statement that a buffer is most effective in the range of 1 pH unit above and below its pKa. The explanation for this apparent paradox is the large reservoir of CO2 in blood. Its rapid equilibration with H2CO3 results in the formation of additional H2CO3. It is useful in clinical medicine to have a simple expression for blood pH in terms of dissolved CO2, which is commonly monitored along with other blood gases. We can define a constant, kH, which is the equilibrium constant for the hydration of CO2 to form H2CO3. The concentration of water is so high, 55.5 m, that dissolving CO2 doesn't change H2O appreciably, so H2O is made part of the constant kH. Then, to take the CO2d reservoir into account, we can express H2CO3 as KHCO2d and substitute this expression for H2CO3 in the equation for the acid dissociation of H2CO3. Now, the overall equilibrium for dissociation of H2CO3 can be expressed in these terms. We can calculate the value of the new constant combined and the corresponding apparent pK or P combined from the experimentally determined values of kH, 3.0 times 10, 3 m, and Ka, 2.7 times 10 4 m, at 37 degrees Celsius. In clinical medicine, it is common to refer to CO2d as the conjugate acid and to use the apparent, or combined, pKa of 6.1 to simplify calculation of pH from CO2d. The concentration of dissolved CO2 is a function of pCO2, which in the lung is about 4.8 kilopascals kPa, corresponding to H2CO3, 1.2 m. Plasma is normally about 24 m, so is about 20, and the blood pH is 6.1 plus log 20 almost equals 7.4. Untreated diabetes produces life-threatening acidosis. Human blood plasma normally has a pH between 7.35 and 7.45, and many of the enzymes that function in the blood have evolved to have maximal activity in that pH range. Enzymes typically show maximal catalytic activity at a characteristic pH, called the pH optimum, Fig. 2 to 22. On either side of this optimum pH, catalytic activity often declines sharply. Thus, a small change in pH can make a large difference in the rate of some crucial enzyme-catalyzed reactions. Biological control of the pH of cells and body fluids is therefore of central importance in all aspects of metabolism and cellular activities, and changes in blood pH have marked physiological consequences, as we know from the alarming experiments described in Box 2 to 1. Figure 2 to 20. Box 2. Rabbit, don't. At home. I wanted to. J.B. A century. In individuals with untreated diabetes mellitus, the lack of insulin, or insensitivity to insulin, depending on the type of diabetes, disrupts the uptake of glucose from blood into the tissues and forces the tissues to use stored fatty acids as their primary fuel. For reasons we describe in detail later in the book, see Fig. 23 to 31, this dependence on fatty acids results in the accumulation of high concentrations of 2-carboxylic acids, beta-hydroxybutyric acid and acetoacetic acid, a combined blood plasma level of 90 mg/100 ml, compared with log mg/100 ml in control, healthy, individuals. Urinary excretion of 5g/24 hr, compared with 125 mg/24 hr in controls. Dissociation of these acids lowers the pH of blood plasma to less than 7.35, causing acidosis. Severe acidosis leads to headache, drowsiness, nausea, vomiting, and diarrhea, followed by stupor, coma, and convulsions, presumably because, at the lower pH, some enzymes do not function optimally. When a patient is found to have high blood glucose, low plasma pH, and high levels of beta-hydroxybutyric acid and acetoacetic acid in blood and urine, diabetes mellitus is the likely diagnosis. Other conditions can also produce acidosis. Fasting and starvation force the use of stored fatty acids as fuel, with the same consequences as for diabetes. Very heavy exertion, such as a sprint by runners or cyclists, leads to temporary accumulation of lactic acid in the blood. Kidney failure results in a diminished capacity to regulate bicarbonate levels. Lung diseases, such as emphysema, pneumonia, and asthma, reduce the capacity to dispose of the CO2 produced by fuel oxidation in the tissues, with the resulting accumulation of H2CO3. Acidosis is treated by dealing with the underlying condition, administering insulin to people with diabetes, and steroids or antibiotics to people with lung disease. Severe acidosis can be reversed by administering bicarbonate solution intravenously. Worked example with bicarbonate. Why does solution where A is increased? Summary 2.3 Buffering against pH changes in biological systems. A mixture of a weak acid or base and its salt resists changes in pH caused by the addition of H+ or OH. The mixture thus functions as a buffer. The pH of a solution of a weak acid or base and its salt is given by the Henderson-Hasselbalch equation. In cells and tissues, phosphate and bicarbonate buffer systems maintain intracellular and extracellular fluids at their optimum physiological pH, which is usually close to 7. Enzymes generally work optimally at this pH. Medical conditions that lower the pH of blood, causing acidosis, or raise it, causing alkalosis, can be life-threatening. 2.4 Water as a reactant. Water is not just the solvent in which the chemical reactions of living cells occur, it is very often a direct participant in those reactions. The formation of ATP from ADP in inorganic phosphate is an example of a condensation reaction in which the elements of water are eliminated. Fig. 2-23. The reverse of this reaction, cleavage accompanied by the addition of the elements of water, is a hydrolysis reaction. Hydrolysis reactions are also responsible for the enzymatic depolymerization of proteins, carbohydrates, and nucleic acids. Hydrolysis reactions, catalyzed by enzymes called hydrolyses, are almost invariably exergonic. By producing two molecules from one, they lead to an increase in the randomness of the system. The formation of cellular polymers from their subunits by simple reversal of hydrolysis, that is, by condensation reactions, would be endergonic and therefore does not occur. As we shall see, cells circumvent this thermodynamic obstacle by coupling endergonic condensation reactions to exergonic processes, such as breakage of the anhydride bond in ATP. You are, we hope, consuming oxygen as you read. Water and carbon dioxide are the end products of the oxidation of fuels such as glucose. The overall reaction can be summarized as the metabolic water formed by oxidation of foods and stored fats is actually enough to allow some animals in very dry habitats, gerbils, kangaroo rats, camels, to survive for extended periods without drinking water. The CO2 produced by glucose oxidation is converted in erythrocytes to the more soluble, in a reaction catalyzed by the enzyme carbonic anhydrase. In this reaction, water not only is a substrate, but also functions in proton transfer by forming a network of hydrogen-bonded water molecules through which proton hopping occurs. Fig. 2-14. Green plants and algae use the energy of sunlight to split water in the process of photosynthesis. In this reaction, A is an electron-accepting species, which varies with the type of photosynthetic organism, and water serves as the electron donor in an oxidation-reduction sequence, see Fig. 20-14, that is fundamental to all life. Figure 2-23 Participation of water in biological reactions. ATP is a phosphoanhydride formed by a condensation reaction, loss of the elements of water. Summary 2.4 Water as a reactant. Water is both the solvent in which metabolic reactions occur, and a reactant in many biochemical processes, including hydrolysis, condensation, and oxidation-reduction reactions.